Hello everyone, welcome to the Animal Zone with Professor Monzon. Today we will talk about another activity of life, circulation, which is really a continuation from the last lesson on homeostasis, as I hope will become very evident soon. We will follow the outline from Hickman's Integrated Principles of Zoology, Chapter 31. First, I will just introduce some general ideas about circulation, and then we'll talk about the internal fluid environment, then about blood in particular, and then some general ideas about vertebrate circulatory systems. Let's start with these questions. Why do most animals need a circulatory system? And which animal groups lack a circulatory system? To answer the second question, you'll have to go back into the previous unit, the diversity of animal life, in which we discussed several animal groups that completely lack a circulatory system. But to answer the first question, why do most animals need a circulatory system? Well, let's see what the circulatory system does. The circulatory system transports nutrients, wastes, and respiratory gases to and from all tissues of the body. The circulatory system also transports hormones to target tissues. The circulatory system responds to disease and injury. The circulatory system distributes and exchanges water and electrolytes for osmoregulation. And the circulatory system conserves or dissipates heat for thermoregulation. So I hope that you see clearly how the circulatory system is one of the ways that many organisms maintain homeostasis. Body fluids can be divided into different compartments. There's the intracellular compartment and the extracellular compartment. The intracellular compartment is basically what's inside of cells. When we discuss the intracellular fluid, it's pretty much synonymous with cytoplasm. And then there's extracellular fluids. These are fluids that are outside of the cells, that buffer the cells from harsh physical and chemical changes outside the body. In animals like ourselves that have closed circulatory system, like the vertebrates or the annelids, the extracellular fluid can be further divided into two kinds: the interstitial fluid or tissue fluid and the blood plasma. So if we look at this illustration here that conceptualizes this, that body fluids can be compartmentalized into the intracellular fluid, aka cytoplasm, and also the extracellular fluids, which can be the tissue fluids and the blood plasma. Now, importantly, all exchanges with the environment occur across the plasma compartment. Think about it. When we eat food, that food that is traveling down the alimentary canal is going to interact with our blood plasma in the capillaries of the gut, and the food will be absorbed and enter into the blood plasma. Similarly, it is the blood plasma as it gets filtered in the kidney where the excretory wastes that are in the blood plasma will be transferred out of the blood plasma and be concentrated in the bladder for elimination as urine. So all exchanges with the environment occur across the blood plasma. The plasma, the interstitial fluids, and the intracellular fluids are mostly water. This is why we say that animal bodies are made up mostly of water. In fact, depending on the animal, animals range from being 70 to 90% water. Us humans, we are about 70% water by weight. So think about how much you weigh. 70% of that is just water. About 50% of your weight is cell water, so intracellular fluid. aka cytoplasm. About 15% of your body weight is interstitial tissue fluid. Those are just fluids that are outside of cells and are also outside of your blood vessels that are just bathing all your tissues. And only about 5% of your total body weight is blood plasma. Now, body fluids can contain many inorganic and organic substances. The organic substances can be proteins or sugars, hormones, but the inorganic substances are typically ions. These are negatively or positively charged small molecules. So you see here in this graphic that the extracellular fluid, in this case blood plasma, has certain concentrations of cations or positively charged ions and other concentrations of anions or negatively charged ions. The intracellular fluid has some of the same cations and anions but at very different concentrations. For example, sodium is at a very high concentration in the blood plasma, but at a very low concentration in the intracellular fluid. Conversely, potassium is at a very high concentration in the intracellular fluid and a very low concentration in the blood plasma. These differences in ion concentrations are drastic, and remember that ions can flow across the plasma membrane. So then what keeps these ion concentrations so different inside and outside the cell? Well, those ion concentrations are very important to keep separate, and they are primarily kept the way they are by active ion pumps. So make sure that you understand this, because I can ask several questions here. For example, what are the primary extracellular electrolytes? Or I can ask specifically, what are the primary extracellular cations? Or what are the primary intracellular electrolytes? Also note that protein is much more abundant inside of the cells than it is outside of the cells. Let's talk about blood for a moment. Invertebrates that lack a circulatory system also lack blood, of course. Some examples of such animals are flatworms or cnidarians. And if you think about it, they don't even need circulatory systems. These animals are so thin that the functions of a circulatory system are unnecessary. Food is just dispersed throughout the animal by diffusion, and also oxygen is absorbed by the animal from the outside by diffusion. So these animals are so thin, they don't need a circulatory system. Some animals, like arthropods, have a type of blood called a hemolymph, but they have an open circulatory system. So they have some blood vessels, but they kind of just dump out into this body cavity, and so it's not true blood as we think of blood being the main fluid of a closed circulatory system, like it occurs in vertebrates and annelids. In those animals, true blood is contained in blood vessels and is completely separated from the interstitial fluids. Now, in invertebrates like ourselves, blood is a complex liquid tissue that is composed of plasma, a liquid, but also cells. So let's talk about that liquid component of the blood. If we were to separate the liquid from the cellular components of the blood, most of your blood is made out of that liquid component called plasma. About 55% of your blood is plasma. Now, about 90% of that 55% is just water. The rest is a bunch of dissolved stuff like plasma proteins, glucose, amino acids, electrolytes, enzymes, antibodies, hormones, wastes, and gases also. The gases are primarily oxygen, carbon dioxide, and nitrogen. Now, if we just focus on the plasma proteins mentioned here, they come in three kinds: the albumins, the globulins, and fibrinogen. Most of your plasma proteins are albumins, about 60% of them. These function mainly in maintaining osmotic equilibrium with the cells. About 35% of your plasma proteins are in the family of globulins, and some of the globulins are special and they're called immunoglobulins, and they function in acquired immunity. And then the third main type of plasma protein is fibrinogen, and this is a very large protein that is just hanging around in the cell waiting to be activated in case a clot needs to form in the case of an injury. And I'll discuss that in just a moment. Now, the rest of your blood, the remaining 45%, is made out of cells. So this is why we say that blood is a tissue, because it's made out of different types of cells with different functions. There are the erythrocytes, which are the red blood cells. These cells contain hemoglobin to transport oxygen and carbon dioxide. Then there are the leukocytes, which come in various flavors, but generally we call them leukocytes or white blood cells, and these are the scavengers and the defenders against foreign materials. So they're part of the immune system. And then there are the thrombocytes, which in mammals we call platelets because they're not really cells, they're more like fragments of cells, but in other vertebrates they are intact cells, which are called thrombocytes. Yeah, so in mammals, they're cell fragments. In other vertebrates, they're intact whole cells. But whether they're fragments or intact cells, thrombocytes function in blood clotting. So let's discuss blood clotting. This is really an important part of homeostasis, right? If there's an injury, we want to restore to its original state. Blood coagulation is a complex cascade of chemical reactions that produce a tangled network of fibrin to stop bleeding at a site of injury. Let's look at this illustration. So here we have a blood vessel that has been injured. Okay, so this is the wall of the blood vessel which has been ruptured, and so platelets, which are just floating around in the blood, are going to collect at the wound site. They're going to recognize the wound and congregate there. The platelets are going to release platelet thromboplastin and other platelet factors. And even the wounded tissue itself will release tissue thromboplastin and other tissue clotting factors. Those two will combine with calcium that's in the blood and create what's called the prothrombin activator. Now, prothrombin is one of those proteins that's just hanging out in the blood. It's an enzyme, but it's inactive. And so prothrombin activator is going to do, guess what? Activate the prothrombin. And so it's going to activate the prothrombin and turn it into thrombin, which is an enzyme that is now active. Remember, fibrinogen is also one of those large proteins that's just hanging out in the blood. However, it is inactive, and thrombin is going to activate fibrinogen and turn it into fibrin, which is an insoluble, tough, fibrous protein, and that is what makes the actual blood clot. So here in this electron micrograph of a blood clot, you see the strands of fibrin creating this meshwork that is literally trapping blood cells from continuing to escape. Now, I quickly mentioned these clotting factors here, the platelet factors and the tissue factors. There are many of them. The idea here is that the clotting factors are normally inactive until they're activated by a previous factor in the sequence. And there are many of them, at least 13 different but genetically similar clotting proteins have been identified. Now, what do you think may happen if there is a mutation causing a deficiency in a clotting factor. These are the kinds of causes for different diseases of the circulatory system. Now, I want to go quickly through this, but the idea here is that I want you to recognize some important differences among the circulatory systems of various vertebrate groups. For example, in the fishes, remember that they have a two-chambered heart. Here in the amphibians, they have a three-chambered heart with two atria and one ventricle, and in the mammal, there's a four-chambered heart with two atria and two ventricles. There are other important features of the circulatory systems that I want you to study out in this figure in the surrounding text. So here, if we zoom into the heart, we see here a comparison of the frog heart and a human heart. And again, the idea is the frog heart has two atria and one ventricle. And so both oxygenated and deoxygenated blood are going to mix in the ventricle. Okay? We have a more efficient compartmentalization of the heart in mammals with two atria and two ventricles that are separated by the septum, thereby keeping the oxygenated blood completely separate from the deoxygenated blood, making the mammalian circulatory system more efficient. I do want you to know the anatomy and flow of blood of the human heart. Here's how I learned it, and here's how I would encourage you to learn it. Draw a heart, just like this. Draw a heart, separate it in two. Draw a line right to separate it, the left and the right. Then you draw another line horizontally to separate the left into a left atrium and a left ventricle, and the right into a right atrium and a right ventricle. So now you have four chambers of the heart. Now there are blood vessels coming into the atria. I want you to know what those are. And there are blood vessels coming out of the ventricles. I want you to know which those are. Then there are different valves that prevent the backflow of blood from the ventricle to the atria. I want you to know which those are. They're in this illustration. And there are valves in the arteries coming out of the ventricles that prevent backflow from the arteries to the ventricles, and those are here, and I want you to know which those are. Okay, so it's real simple. Four chambers, four main blood vessels, and four valves. Here's some vocabulary. We call the contraction of the heart systole, and we call the relaxation of the heart diastole. Systole and diastole. So the contraction and the relaxation of the four chambers of the heart is not all in unison, and they're also not random. What happens is that the two atria are synchronized, they contract together, and while they're doing that, the two ventricles relax together. So that's what we see here. We call that atrial systole and ventricular diastole. So when the atria contract, you're going to squeeze blood into the ventricles, and so now the ventricles are full of blood and the atria are empty of blood. So what happens next is that the atria are going to relax so they can receive more blood, and the ventricles are going to contract so they can force out the blood. Okay, so that's what we have here, atrial diastole and ventricular systole. And so that's really what causes the dub 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 of the heart. The first dub is the atrial contractions, and the second one are the ventricular contractions. Notice that the walls of the ventricles are much more muscular than the walls of the atria. Why do you think that is? Think about it. How far does the blood need to go after leaving the atria versus how far does the blood need to go after leaving the ventricles? Now, what gets your heart going? Well, itself, because the heart is myogenic, the vertebrate heart at least. The vertebrate heart is myogenic. That means it self-starts and self-excites. What does that mean? Well, we're really just scratching the surface here of the heart. But I'll just say this. In the amniote heart, there's a specialized bundle of muscle fibers here in the atrium called the sinoatrial node. We also call it the pacemaker. And it is the pacemaker which initiates each heartbeat. Essentially, it's an action potential that is going to spread across the heart. The electrical activity coming from the sinoatrial node is going to spread across the muscular wall of the atrium. After a slight delay, is going to reach a secondary pacemaker called the atrioventricular node, or the AV node right here. And that electrical activity is going to continue spreading through the bundle of His, through the left and right branching bundles, and ultimately ending at the Purkinje fibers at the apex of the ventricles. So you see there's a delay between the contraction here and the contraction here, which is why it makes perfect sense, because the atria need to contract first and then the ventricles. Now, although the heart is myogenic, meaning it self-starts and self-excites, there is also some external regulation of the heart. We have in the medulla of our brain a cardiac center, which provides external regulation to the heart via two sets of nerves that terminate in the SA node, the sinoatrial node. Those are the sympathetic nerves, which increases the heart rate, and the parasympathetic vagus nerves, which decrease the heart rate. The cardiac center itself receives some input information from pressure receptors in your blood vessels. These are receptors that are scattered throughout your blood vessels that are monitoring your blood pressure. And also chemical receptors, which are scattered throughout your blood vessels that are monitoring how much CO2 is dissolved in your blood and the pH of your blood. If you hold your breath for a long time, at some point you will be unable to hold it anymore, and you will go, what is it that makes you do that? Is it a lack of oxygen? Actually, is a surplus of CO2. Your blood vessels have chemical receptors that are sensitive to how much CO2 there is, and when there is too much CO2, it will make you breathe more and will make you your heart beat faster. Again, same thing when you are running or doing some other kind of strenuous exercise. Your heartbeat will pump faster, not because you need more oxygen, but because you need to expel more CO2 than normal. So that cardiac center in your brain uses all of this information to adjust the heart rate and the stroke volume, which together result in the cardiac output. It's really the simple product. So the cardiac output is the product of the stroke volume and the heart rate. So let's end with some of these review questions. But I hope you enjoyed this lesson on the circulatory system. Hope your heart is doing well. Continue washing your hands frequently, and always be strong and courageous.